Welcome to another MedicoMedics lecture. This time we're diving into genetics. You can also check out our full series on microbiology, cell biology, and more, all right here on the channel. This full course covers 13 chapters from DNA and Mendel all the way to genomics, cancer genetics, and beyond. So let's get to it. Welcome to another lecture by MedicoMedics, Learning Made Easy. Genetics, Chapter 1, Introduction to Genetics. In this lecture, we will discuss what is genetics, briefly touch upon the history of genetics, the discovery of DNA, why genetics is important in medicine, some key concepts, and end with a summary. Now, genetics is the science of heredity and variation in living organisms. Focus areas include genes, which are the units of heredity, DNA, the blueprint of life, and chromosomes. These carry the genetic material. Why is this relevant? Because it explains biological diversity, and it links traits to their hereditary origins. So an example of this is, why do some people have curly hair while others don't? That's an example of genetics at work. Now, before modern science, people thought that traits blended together like mixing colors. But it wasn't until the discovery of cells where we got our first clues to start understanding heredity correctly. Because, for example, the blending inheritance theory couldn't explain trait reappearance. And pangenesis was the idea that small particles from the body would transfer traits to our offspring. And it implied that acquired traits could be inherited. And this was later disproven. Only after the discovery of cells, with the technological advancements of microscopy, and when we get to Gregor Mendel, who is called the father of genetics, did we start understanding heredity. So Gregor Mendel, called the father of genetics, what he did was he experimented with pea plants, and he observed patterns of inheritance in traits, for example, tall versus short plants. And that led him to formulate two laws. First, the law of segregation. So each parent passes one copy of a gene. He also formulated the law of independent assortment. So traits are inherited independently. So, for example, tall plants crossed with short plants produce tall offspring, but short plants can appear in the next generation. So, following from Mendel's discoveries, we found that traits actually don't blend like they thought before. Instead, they are passed down like in patterns, and it's like flipping a coin. And then, of course, with the discovery of DNA, everything accelerated. Now, Friedrich, in 1869, discovered the nuclein, later known as DNA, and he did that in white blood cells. In 1952, the Hershey-Chase experiment proved DNA, not proteins, is the genetic material using viruses and radioactive labeling. Further on, 1953, Watson, Crick, and Franklin used X-ray diffraction to describe the DNA double helix structure. And the structure of DNA, therefore, as we know it now, is a double helix, so a spiral staircase, and you can see it illustrated here. And it is made of sugar, phosphate, and nitrogen bases. And then you find A, T, C, G pairings, where you have adenine, thymine, cytosine, and guanine. So DNA is like a twisted ladder made of molecules. It carries the instructions for how your body grows and works. And further, moving into modern genetics, we've had the Human Genome Project that went on from 1990 to 2003, where we mapped all human genes. And this means that, similar to creating a GPS for the human body, it helps us to develop better treatments for diseases. And of course, it enabled breakthroughs in medicine and technology. Some of these technologies could be CRISPR, which is a gene editing tool. So an example is we use gene therapy for conditions like muscular dystrophy, where modern genetic tools are used. And this gene therapy is becoming more and more and more popular now. So why genetics is important in medicine? Because first of all, diagnostics. It helps us identifying genetic causes of diseases. For example, sickle cell anemia. Personalized medicine. So tailoring treatments based on DNA. For example, targeted cancer therapy. Preventive measures. So predicting disease risk, like BRCA mutations for breast cancer. Now, some key concepts in genetics include the gene, which is the unit of heredity, and it codes for proteins. Then we have chromosomes. These structure organizing DNA in cells. And we have 23 pairs in humans, and in total, 46. Mutations, so change in DNA sequence that can affect health. An example is a mutation in the hemoglobin gene, leading to sickle cell disease. So a way to think about it is that genes are like instructions. Chromosomes are the books that hold these instructions, and mutations are typos in those instructions. So in summary then, genetics studies how traits and diseases are inherited. It's crucial in medicine, agriculture, and technology. Historical milestones include Mendel's laws and the discovery of DNA. And modern genetics drives personalized medicine and new treatments. That's the end of the first chapter. Continue now to chapter 2. Welcome to another lecture by MedicoMedics, Learning Made Easy. Genetics Chapter 2, Cellular Basis of Genetics. In this lecture, we will present an overview of DNA structure, chromosomes, the process of mitosis and meiosis, errors in cell division, briefly mention ribosomes and protein synthesis, and end with a summary. Now, the basic unit of life is the cell, and it contains the genetic material. We have a nucleus, which is the control center of the cell where the DNA resides. And some organelles involved in genetics include nucleus, which contains the chromosomes, and ribosomes. These read genetic instructions in order to make proteins. Now, DNA is like a twisted ladder with steps made of chemical pairs. And you can see an illustration here of DNA. Now, DNA stands for deoxyribonucleic acid. The shape is a double helix, and its components include a sugar phosphate backbone and nitrogen bases, A, T, C, and G, where A is adenine, T is thymine, C is cytosine, and G is guanine. And these are paired together. So A is paired with T, and C is paired with guanine. And this is what tells your body how to work. The arrangement order. So the function of DNA is it's a blueprint for making proteins. And it's passed from parents to offspring. So we can mention that this sugar phosphate backbone, what it does is that the phosphate group provides structural support by linking to the sugar molecule of the next nucleotide. And deoxyribose sugar is a 5-carbon sugar that acts as the backbone to which the phosphate group and nitrogenous bases are attached. And the nitrogenous base, so these are the variable components of the nucleotides. And these include the 4 types mentioned here: adenine, thymine, cytosine, and guanine. And these bases, right, as we just mentioned, encode the genetic information through specific pairings. Now, chromosomes are thread-like structures made of DNA and proteins found in the nucleus. These organize and store genetic information, ensuring accurate DNA replication and gene expression. Now, humans have 23 pairs of chromosomes, so 46 chromosomes in total. 22 of these pairs are autosomes, so these are non-sex chromosomes, and these generally control our general body traits. We then have 1 pair of sex chromosomes, so these determine biological sex. If it is XX, we have female. If it is XY, then we have male. Now, DNA is tightly coiled around histone proteins, and this forms something called chromatin, which further condenses into chromosomes during cell division. And this compact structure ensures DNA fits within the cell nucleus while allowing controlled gene expression. Now, let's talk about cell division, also known as mitosis. So mitosis is the process by which a cell divides to create 2 identical daughter cells. And this is high yield, 2 identical daughter cells. Why it's important is because we will talk about the process of meiosis. And one way they differ, or it differs from mitosis, is the identical part of it all. But it will make sense as we move along. Now, the phases of mitosis include prophase, metaphase, anaphase, and telophase. We also have, we may add it here, something called cytokinesis at the end. So in prophase, the chromosomes condense. In the metaphase, these chromosomes align in the center. During anaphase, these chromosomes separate, each to a side. So these so-called centromeres move apart, and half of the chromosomes go to one pole, the other half to the other pole. Then we enter the telophase, and this is where a nuclear membrane forms around these separated chromosomes. So you're beginning to see that it's becoming 2. So during telophase, 2 new nuclei form. And the general purpose of mitosis is to generate growth repair and replacement of cells. For example, our skin cells divide through mitosis to heal when we get a cut. So mitosis is how your body makes new cells to grow or replace old ones. Now then, after mitosis, we have another type of cell division, which is meiosis. And meiosis is the cell division that produces gametes, so sperm and egg cells. And it's produced with half the number of chromosomes. The phases include 2 rounds of division. So we have meiosis 1 and we have meiosis 2. And the result of meiosis is 4 genetically unique cells with 23 chromosomes each. The purpose of this is reproduction and genetic diversity. So example, meiosis ensures you get half your DNA from your mom and half from your dad. Now here you can see an illustration where we have meiosis 1 and meiosis 2. So in meiosis 1, which is the reductional division is occurring here, we have homologous chromosomes separating. So in humans, there are 23 pairs of homologous chromosomes, so 46 in total, in a diploid cell. Each pair consists of 1 chromosome from the mother and 1 from the father. Now, during prophase 1, crossing over occurs, where these chromosomes exchange genetic material and introducing genetic diversity. So during prophase 1, and this is high yield, you have the exchange of genetic material and genetic diversity. Now, in metaphase 1 and then following to anaphase 1, this is where they are aligned and then they become separated. So in anaphase 1, homologous chromosomes, not sister chromatids, are pulled apart to opposite poles. This is similar to mitosis. And at the end of meiosis 1, the diploid cell divides into 2 haploid cells, each with 23 chromosomes. Now, these 23 chromosomes are still in their duplicated form, joined at the centromere. Then we have meiosis 2, and this is called the equational division, or where equational division is occurring. So this is where sister chromatids separate. So meiosis 2 is actually very similar to mitosis. In anaphase 2, the sister chromatids of each chromosomes are pulled apart to opposite poles. And the end result of meiosis 2 is that each of the 2 haploid cells from meiosis 1 divide again, producing in total 4 haploid gametes, so sperm or egg cells in humans, each with 23 single chromosomes. Now, just some key clarifications here. So crossing over happens only in prophase 1 during meiosis 1, not after. And meiosis 2 does not involve chromosome duplication. It simply separates the sister chromatids of the already half chromosome set. So again, the end product, 4 genetically unique haploid cells, each with 23 chromosomes ready for sexual reproduction. So put into a table the difference between mitosis and meiosis. So the purpose of mitosis is growth and repair, and the purpose of meiosis is reproduction. The number of cells in mitosis is 2 identical cells, And in meiosis, it's 4 unique cells. So the number of cells is different. Here they are identical in mitosis, and they are unique in meiosis. The chromosome count, same as parent, 46. And in meiosis is half of parent, so 23. So mitosis is essentially for making more of the same cell, while meiosis is for making cells with half the DNA for reproduction. And that's the key difference between mitosis and meiosis. Hope that's clear. Now let's talk about errors in cell division. So we can have non-disjunction, which is a failure of chromosomes to separate properly during meiosis. So it's an error in cell division where chromosomes indeed fail to separate properly during either meiosis 1 or 2. And this results in gametes, so sperm or eggs, with an abnormal number of chromosomes. Now, if you have a meiosis 1 failure, it means that homologous chromosomes do not separate. If we have a meiosis 2 failure, it's that sister chromatids do not separate. And consequences of non-disjunction. So it can lead to abnormal chromosome numbers. For example, Down syndrome, which is known as trisomy 21. It means we have 3 copies of chromosome 21. We can have Turner syndrome, where we are missing 1 X chromosome in females. It's also known as XO. So Turner syndrome, we're missing 1 X chromosome in females. Trisomy 21, or Down syndrome, we have 3 copies of chromosome 21. So an additional copy. We also have another one known as Klinefelter syndrome, which is XX and Y, where we have an extra X chromosome in males. Because males have XY chromosomes, right? So we're talking about a male here. But we have an additional X chromosome. Now, mutations, so changes in the DNA during replication, can be harmless, harmful, or beneficial. Now, the consequences or the impact of these issues is that they can develop developmental delays, physical abnormalities, and even reproductive issues. And yeah. Now let's just briefly mention ribosomes and protein synthesis. So ribosomes are organelles that assemble proteins using instructions from DNA. So the process is DNA transcribes into RNA. And this process is known as transcription. And it transcribes into a messenger RNA. So you will see M and then RNA. And then ribosomes, these organelles, translate this RNA into proteins. And proteins are essential for building and maintaining our body. So you can think of it like ribosomes use RNA instructions like a chef uses recipes to cook meals. And in summary then, DNA is the blueprint of life stored in chromosomes within the nucleus. Mitosis and meiosis are the processes that ensure genetic material is copied and passed on. Errors in cell division can lead to genetic disorders. Ribosomes are essential for turning genetic instructions into proteins. And that's the end of chapter 2. Continue now to chapter 3. Welcome to another lecture by MedicoMedics, Learning Made Easy, Genetics Chapter 3, Molecular Mechanisms of Inheritance. In this lecture, we will repeat and give you an overview over DNA replication, transcription, translation, and end with a summary. Now, DNA contains the instructions for building and maintaining an organism. Key processes include replication. This is where DNA is copying itself before cell division. Another one is transcription. This is where DNA is converting to RNA, more specifically, messenger RNA. Then we have translation. This is where the RNA is used to make proteins. And regulation ensures only the required genes are active. So DNA is like a library of instructions, and replication makes a backup copy, while transcription writes the notes. And translation then uses these notes to make proteins. Now, DNA replication, basics. So what happens here? Well, DNA makes a copy of itself, and this occurs in the S phase of the cell cycle. And this is an illustration of the cell cycle. And the synthesis phase is illustrated here in green. Now, the key enzymes involved are helicase, and this enzyme unwinds the DNA helix. We then have DNA polymerase. This adds new bases to the growing strand. And then we have ligase, which seals gaps in the DNA backbone. The outcome? Two identical DNA molecules, each with one old strand and one new strand, also called semi-conservative replication. Now, the steps in detail. So we have the terms initiation, elongation, and termination. So during initiation, helicase opens the double helix at replication origins. During elongation, DNA polymerase adds complementary bases, A, T, so adenine and thymine, CG, cytosine and guanine. And then towards termination, ligase seals the DNA, completing the process. Now, mutations can occur if proofreading mechanisms fail. For example, an error in replication can lead to conditions like sickle cell anemia if not corrected. So DNA makes a near-perfect copy of itself, but mistakes can happen, and this sometimes lead to genetic diseases. Now let's repeat the process of transcription, where we are converting DNA to RNA. So what happens? DNA is transcribed into messenger RNA, and this takes place in the nucleus. Key players here are RNA polymerase, which copies DNA into RNA. We then have promoter regions, so the DNA region signaling where transcription begins. So to the question, where does transcription begin? It is in the region called the promoter region. Then we have two other important concepts like intron and exons. So non-coding are introns and coding regions are called exons. The important steps included initiation, where RNA polymerase binds to this promoter region. Then we have elongation, where RNA polymerase synthesizes RNA. And then we have termination, where RNA polymerase releases the RNA strand. And a way to remember is that transcription is like writing down instructions from a book. so the DNA, onto a notepad, which would be the RNA. And then this RNA can leave the nucleus and then through the process of translation start producing proteins. Now then, translation, the making of proteins. So what happens? Messenger RNA is translated into a protein, and this takes place in the cytoplasm at ribosomes. Key players here include messenger RNA, so it's the messenger with the instructions. We have transfer RNA, which brings amino acids to the ribosomes. If you look at the illustration here, you can see we have the messenger RNA. So its primary structure. We have the ribosome itself. We have the transfer RNA bringing amino acids. And then the machinery then assembles this to make proteins. Steps here include, again, initiation. So ribosome binds to the messenger RNA. These two. Elongation. So transfer RNA delivers this amino acid, forming a chain. And then termination. A stop codon signals the end. When it's done, it signals stop, and the protein is then released. So translation is like using a recipe, mRNA, to assemble a meal, which would then be the proteins. And these proteins are the building blocks of your body. Now that you've come this far, indeed, there are a lot of new terms, a lot of different steps, and it's easy to be confused. So we will provide a mid-summary here in this lecture to try and contextualize for you all the different parts that we just went through. Now we have something called the central dogma of molecular biology. So ideally going from DNA to proteins. Now the process of genetic expression begins with DNA replication. It's ensuring that genetic material is faithfully copied before cell division. Okay, so we have DNA replication to begin with. And this starts with the enzyme helicase, if you remember from our previous slides. And this enzyme is responsible for unwinding this double helix, creating something called a replication fork. Then we have DNA polymerase. Okay, it's not RNA polymerase, that comes later. DNA polymerase adds a complementary nucleotides to each of these original strand, synthesizing the new DNA. Now, since DNA strands run in opposite directions, replication occurs differently on each. The leading strand is synthesized continuously, while the lagging strand is made in short Okazaki fragments. And these fragments are later joined by ligase, which seals the DNA backbone and completes replication. Okay, so we've had DNA replication happening. Check. Now, once the DNA is replicated, gene expression begins with transcription, where a gene's DNA sequence is now copied into messenger RNA. All right? After that, RNA polymerase, not DNA polymerase, but RNA polymerase, binds to the promoter, a specific DNA sequence signaling the start of transcription. So it binds to this specific place, and that's the kickoff of transcription. Now, this marks the initiation phase, right? Phase 1, initiation. Now, after that, we had step 2, which was elongation. So RNA polymerase moves along the DNA, adding complementary RNA nucleotides. So adenine, uracil, cytosine, and guanine. If you remember, in DNA, we had the pairing of adenine to thymine and cytosine with guanine. But when we're talking about RNA, we have the pairing of adenine with uracil and cytosine with guanine. Now, once this reaches a termination signal, then transcription ends. And the newly formed pre-messenger RNA undergoes processing. Now, this includes removing introns, which are non-coding regions, and splicing together exons, which are coding sequences, forming the final mRNA. All right? Now, this processed mRNA now leaves the nucleus and enters into the cytoplasm. And this is where translation starts. So up till this point, we were in the nucleus of the cell. Now we're in the cytoplasm. And in the cytoplasm, where translation starts, this is where the RNA message, right, from the messenger RNA is now translated or converted into a protein. And this process occurs in the ribosomes. This is the organelle, the ribosome, which reads the mRNA sequence in 3-letter codons. So during initiation, a transfer RNA, tRNA, carrying methionine, or methionine, I think it's called, binds to the ribosome at the start codon. And this start codon is abbreviated AUG. During elongation, more transfer RNAs bring amino acids, matching their anticodons to the mRNA codons, while the ribosome links the amino acids together to form a growing polypeptide chain, so a chain of amino acids. And then finally, in termination, the ribosome encounters a stop codon, promoting the release of this completed protein. So this is a step-by-step journey from DNA replication to transcription, and finally to translation, ensuring that genetic information is accurately preserved and used to build functional proteins which drive all cellular activities. Hope the clarification was clear. We also go into depth in our cell biology course for those who are interested to check that out. So in summary then, DNA replication ensures genetic material is copied. Transcription converts DNA to RNA, and translation turns RNA into proteins. Gene regulation controls when and where genes are active. Errors in these processes can lead to genetic disease or variations. And that's the end of Lecture 3. Continue now to Lecture 4. Welcome to another lecture by MedicoMedics, Genetics Chapter 4, Mendelian Genetics. So in this lecture, we will talk about Mendel's experiments, Mendel's laws, dominant and recessive traits, mention Punnett squares, and talk about exceptions from and applications of Mendelian genetics, and end with a summary. Now, Mendelian genetics is about how traits like height or eye color are passed down from parents to offspring. And it is based on one of his famous pea plant experiments. Now, why is it important? Because it explains inheritance patterns and it forms the basis for modern genetics. So Mendel's experiments, the setup. Why pea plants? Because A, they are easy to grow. They have distinct traits like flower color and seed shape. And they can self-pollinate and or cross-pollinate. And the traits studied by Mendel included seed shape, so round versus wrinkled, seed color, yellow versus green, and flower color, purple versus white. So Mendel crossed tall pea plants with short ones and observed their offspring. And the reason he chose these pea plants is because they are simple and have clear traits, making them easy to study, or making it easy to study how traits are passed along. Now let's begin with his first law, which is the law of segregation. And the law of segregation states that an organism inherits 2 alleles for each trait, one from each parent. And during gamete formation, which is meiosis, these alleles separate so that each gamete, each sperm or egg, carries only one allele for a given trait. So each parent contributes one allele to the offspring, and these alleles can be dominant or recessive. And so an example here, if we have a tall plant expressed in the capital T, which means it's dominant, and we have a short plant in small t, this person will produce an offspring with big T and small t alleles, where the capital T is expressed because it's dominant. So if a tall T plant, right, is crossed with a short plant, small t, the offspring will inherit one allele from each parent. So the offspring will get T and T. Because T is dominant, the plant will be tall. But the recessive T, the little one, is still present and can appear in later generations if the offspring receives another of these small t's, which would make it 2. And that would then be a short plant. Then we have Mendel's second law here, the law of independent assortment. And it's defined as alleles for different traits are inherited independently of each other. So traits like eye color and hair type, etc., they don't always come as a package deal. They're passed on separately. So the inheritance of one trait, like flower color, doesn't necessarily affect the inheritance of another, like seed shape. So crossing plants with yellow, round seeds and green, wrinkled seeds can produce a mix of all combinations. Now let's talk about Punnett squares used to predict inheritance. So a Punnett square is a simple diagram used in genetics to predict the probability of offspring inheriting certain traits based on parental alleles. It visually represents Mendelian inheritance patterns for dominant and recessive traits. So how it works is you write parent alleles along the top and side, here and here. And then you fill in the squares to show possible offspring combinations. So tall and tall, and then you have tall and well, dominant, tall, and short. So TT. You have the short variation with the big one, the dominant. So you get dominant T and T. And then you have recessive with recessive, and you get this one. So what's the genotype here? You can have TT, which is homozygous dominant. So it's going to be a tall person. You have dominant T with recessive. And here you will also have a tall person, but he will carry the recessive gene. But the expressed one is tall. And then you have, again, recessive plus dominant. You have, again, TT. So this is also tall. And then you have recessive and recessive, making it a short person. So the outcome of this would be that there is a 75% chance of being tall and 25% of being short. Now, dominant and recessive traits. So dominant alleles, traits that are expressed when present. So for example, capital T for tall plants. Meaning dominant traits win over recessive traits. But recessive traits can appear if both parents actually pass them on. So when we talk about dominant alleles, these are traits that are expressed when they are present. If we talk about recessive alleles, these are traits that are hidden unless both alleles are recessive. So unless we have that combination of T and T, it will remain hidden. Then we have the concept of homozygous versus heterozygous, or homozygous versus heterozygous. So if it's homozygous, both alleles are the same. So TT or TNT. If it's heterozygous, alleles are different. And so any variation where we have TNT, for example, this is a homozygous. And if the same with two recessives, but all the other combinations were heterozygous. So a child, for example, with one dominant brown eye allele and one recessive blue eye allele will have brown eyes. Now, there are, of course, exceptions to Mendel's laws, like incomplete dominance, so neither allele is completely dominant, resulting in a blend. For example, red and white flowers make pink. We have codominance, so both alleles are fully expressed. For example, blood type A and B. Multiple alleles, so more than two alleles for a trait. For example, blood types A, B, and O. Polygenic traits, so controlled by multiple genes like height, skin, and skin color. Example, a red and white flower cross results in a pink offspring. That's an example of incomplete dominance. So not all traits follow simple dominant recessive rules. Some traits mix or show multiple versions at once. What are some applications of Mendelian genetics? Well, in medicine, predicting risk of genetic disorders like cystic fibrosis. Agriculture, so breeding plants and animals for desired traits like pest-resistant crops. And forensics, using DNA inheritance patterns to solve crimes. Example, genetic testing helps parents understand the risk of passing on diseases like sickle cell anemia. So Mendelian genetics is used in healthcare, farming, and even crime solving to understand how traits and diseases are passed on. So in summary then, Mendel's experiments revealed two key principles. The law of segregation, so genes separate during gamete formation. And law of independent assortment, so traits are inherited independently. Traits can be dominant, recessive, or show variations like codominance or incomplete dominance. Tools like Punnett squares help predict and trace inheritance. And that's the end of Chapter 4. Continue now to Chapter 5. Thank you for listening. Welcome to another lecture by MedicoMedics, Genetics Chapter 5, Non-Mendelian Genetics. In this lecture, we will look at incomplete inheritance, codominance, multiple alleles, polygenic inheritance, environmental influence on genes, mitochondrial inheritance, and end with a summary. Now, in the previous lecture, we talked about Mendelian genetics, and we mentioned the laws of segregation and independent assortment. Now, when we talk about non-Mendelian genetics, we're talking about inheritance patterns that are not explained by Mendel's laws. Now, why is it important? Because it explains complex traits like height, skin color, or diseases. And it reflects how most traits are influenced by multiple factors. So it helps understanding genetic conditions that are not predicted by Mendelian ratios, including, for example, blood types, mitochondrial inheritance, and certain diseases. So some types of non-Mendelian inheritance include, as we mentioned in the previous lecture, incomplete dominance, which is a blend of traits like something, let's say you have a red and a white flower and you get a pink flower. We also have codominance, so both alleles are equally expressed, like AB blood type. Polygenic inheritance, multiple genes controlling a trait like skin color or height. Multiple alleles, so more than 2 allele variations existing, like ABO blood groups. We have sex-linked traits, so genes carried on sex chromosomes like color blindness or hemophilia. Mitochondrial inheritance, traits passed only from the mother via the mitochondria. And epistasis, or epistasis, one gene affects the expression of another. So understanding these patterns is essential in genetics, medicine, and evolution, as most traits result from complex interactions rather than simple dominant recessive rules. So let's briefly look at these one by one. So we have incomplete dominance, and this is where neither allele is completely dominant, resulting in a blended phenotype. For example, crosses between red and white flowers producing pink flowers. A real-life example is snapdragons. If you ever looked at one, you see that red and white flowers could produce pink offspring. Codominance, where both alleles are fully expressed in the phenotype. Example, blood type A and B, so both A and B alleles are expressed equally. An example of this is roan cattle, where red and white hair appear together, not blended. So codominance is like seeing both red and white stripes on a flower instead of them blending into pink. Now we also have multiple alleles, where we have more than 2 alleles existing for a single gene. For example, in blood types, we have allele A, B, and O, and this produces a variation of different combinations. The real-life implication is that, for example, blood transfusions depend on compatible blood types. Polygenic inheritance, so traits controlled by multiple genes. For example, height. This is controlled by many genes, each contributing a small effect. Skin color, so this is determined by several genes interacting with the environment. And characteristics of this include continuous variation, so not just tall or short, but a range. So polygenic traits are like baking a cake with many different ingredients. Each one adds a little to the final result, creating therefore a range of possibilities. And then environmental influence on genes. So essentially, environment affects gene expression. For example, hydrangea color, so soil pH changes flower color. If it's acidic, it's blue. If it's alkaline, it's pink. Our height, for example, so nutrition during childhood influences final height. What are the implications? Well, nature, so genes, and nurture, the environment, work together. So your genes are like a recipe, but the environment is like the ingredients you can use. They can change the final outcome. Another one is mitochondrial inheritance. So inheritance of genes found in the mitochondria passed down from the mother. And characteristic for this is mitochondria contains its own DNA, and disorders here could affect energy production. So for example, we have a variety of mitochondrial diseases. And if you remember from our other lectures, it is in the mitochondria where our main source of energy is actually produced, ATP. An example of due to mitochondrial mutations is Leber's hereditary optic neuropathy. This is vision loss due to mitochondrial mutations. Now, genetic imprinting. So it's defined as the expression of a gene depends on whether it is inherited from the mother or the father. It's an epigenetic phenomenon where only one parental allele, so either mother or father, is expressed, while the other one is silenced. And unlike Mendelian inheritance, where both alleles are expressed equally, imprinted genes show parent-specific expression. Examples in human could be Prader-Willi syndrome, where paternal genes on chromosome 15 are either deleted or silenced, and maternal copies are naturally imprinted, so turned off, leading to developmental issues like obesity and cognitive impairment, etc. We have Angelman syndrome, where maternal genes on chromosome 15 are deleted or silenced, so paternal copies are imprinted, leading to neurological and developmental disabilities, etc. Now, why is genetic imprinting so important? Because it regulates growth and development in early life, and it plays a role in disease inheritance and susceptibility. So errors in imprinting can lead to disorders, cancers, metabolic conditions, etc. Now, anticipation is defined as a genetic disorder becoming more and more severe or appearing more early in subsequent generations. So it is like a genetic snowball. Each generation inherits a larger, more severe version of the condition. Examples of this is, for example, Huntington's disease, where symptoms worsen and appear earlier in offspring due to repeated expansion in the DNA. So repeated sequences in the DNA grow longer with each generation. And what is the concept of epigenetics? So it is defined as changes in gene expression without altering the DNA sequence. Mechanisms include DNA methylation, where there is a silencing of genes, histone modification, so changes how tightly DNA is packaged. Example, stress in early life can turn genes on or off, affecting health later. So epigenetics is like flipping a switch to turn genes on or off without changing the actual wiring. Now, what could be some applications of non-Mendelian genetics? Well, in medicine, we use it to understand complex diseases like diabetes, cancer, schizophrenia. In agriculture, use it to breed plants with desired traits. In forensics, identifying unique genetic markers for solving crimes. An example is studies on polygenic traits help predict risk for heart disease by analyzing many genes together. So in summary then, inheritance patterns include incomplete dominance, codominance, multiple alleles, and polygenic traits. Environmental factors and epigenetics modify how genes are expressed. Applications span from healthcare to agriculture and forensic and beyond. That's the end of Chapter 5. Thank you for listening. Continue now to Chapter 6. Welcome to another lecture by MedicoMedics, Genetics Chapter 6, Genetic Mutations and Disorders. In this lecture, we will give you an overview of genetic mutations, causes of these, types of genetic mutations. Further on, we will talk about chromosomal mutations, genetic disorders, multifactorial disorders, diagnosing and treating these genetic disorders, and end with a summary. Now, what is a mutation? A mutation is a change in the DNA sequence. And there are different types of these. We have harmless mutations, so neutral or silent mutations. We have harmful mutations. These cause genetic disorders or diseases. We even have beneficial mutations. These contribute to, for example, evolution, like resistance to malaria. So mutations are changes in your DNA. Some don't matter, some cause problems, and some can even be helpful. Now, what are some causes of these mutations? We have spontaneous mutations, so errors during DNA replication. We have natural chemical changes, like deamination. We have induced mutations caused by, for example, radiation, like UV rays or X-rays. It can be caused by chemicals like tobacco smoke or benzene, viruses like HPV causing genetic damage. For example, UV radiation from sunlight can cause mutations leading to skin cancer. Now let's talk about different types of genetic mutations, starting with point mutations. Now, point mutations occur when a single nucleotide in DNA is altered, potentially affecting protein function. These mutations can be classified into three main types. We have silent mutation. So it's a change in a nucleotide that does not alter the amino acid sequence due to the redundancy of the genetic code. These mutations usually have no effect on protein function. Then we have missense mutations. So a single nucleotide change results in a different amino acid being incorporated into a protein. So a single nucleotide change results in a different amino acid being incorporated into a protein. So this can affect protein structure and function, sometimes leading to diseases. An example is sickle cell anemia, caused by a mutation in the hemoglobin gene, where glutamic acid is replaced by valine, leading to misshapen red blood cells. Then we have nonsense mutations. So a single nucleotide change creates a premature stop codon. So a single nucleotide change creates a premature stop codon, leading to an incomplete and usually non-functional protein. And this can cause severe genetic disorders. Example, again, sickle cell anemia caused by single base change in the hemoglobin gene. Now moving on to a different type of genetic mutation called frameshift mutations. Now these frameshift mutations occur when nucleotides are inserted or deleted from the DNA sequence, altering the reading frame of the genetic code. Since the genetic code is read in triplets, in codons, this shift changes the entire sequence downstream, often leading to a completely different or non-functional protein. So An example is Tay-Sachs disease or Tay-Sachs disease. I'm not sure about the pronunciation here. It is caused by a frameshift mutation in the hexa gene, this leads to a dysfunctional enzyme that results in the accumulation of harmful substances in the brain, causing neurodegeneration. And frameshift mutations, overall, tend to have more severe consequences than point mutations due to their widespread impact on protein function. Moving on to chromosomal mutations. So chromosomal mutations are structural changes in the chromosomes that can affect an organism's genetic information. These mutations can result from errors during cell division, such as meiosis or mitosis, and can have significant effects on an individual's health and development. Example could be deletion. So a portion of the chromosome is missing or is lost, leading to absence of genetic material. An example is Cri-du-Chat syndrome, caused by a deletion on chromosome number 5, and it leads to intellectual disability and a distinctive high-pitched cry in infants. Then we have duplication, where a segment of the chromosome is duplicated, leading to extra copies of genes. An example is Charcot-Marie-Tooth disease, often caused by a duplication on chromosome number 17, leading to progressive nerve damage. We have inversion, where a chromosome segment is reversed end to end. And since no genetic material is gained or lost, inversions may not always cause noticeable effects, but can disrupt gene function in some cases. And also then we have translocation. So this is where a segment of one chromosome is transferred to another non-homologous chromosome. This can be balanced with no genetic material lost, or unbalanced with extra or missing genes. An example is chronic myeloid leukemia, caused by a specific translocation between chromosomes 9 and 22, forming the so-called Philadelphia chromosome, which leads to uncontrolled cell growth. Now, these mutations can have varying impacts, from minor effects to severe genetic disorders. Some mutations may be inherited, while others occur spontaneously due to environmental factors such as radiation or chemical exposure. Now let's briefly mention some single gene disorders. So these are disorders caused by mutations in a single gene. Examples include cystic fibrosis caused by mutations in CFTR gene, Huntington's disease, where a mutation has occurred with repeated DNA sequences, and sickle cell anemia, a single point mutation in the hemoglobin gene. Chromosomal disorders. These are disorders caused by chromosomal abnormalities, including, for example, Down syndrome, also known as trisomy 21. Essentially, there is an extra chromosome copy of chromosome 21. And this leads to intellectual disability, very distinct facial features, and an increased risk of heart defects. We also have Turner syndrome, also known as monosomy X, where an X chromosome is missing, or we only have one X chromosome, and this only affects females. It's also written out as 45X or XO. And this leads to the typical presentation is short stature, delayed puberty, and infertility. And another one worth of mentioning is Klinefelter syndrome, where you have an extra X chromosome in males. So this one only affects males, and you have an additional X chromosome, And this is written out as 47XXY. And you know that we have a total of 46 chromosomes normally, 2 pairs of 23s. So here we have an extra one, and here we have 45. So one is missing. What is missing? An X chromosome. And this only affects females. So this one only affects males. And Down syndrome, where you have an extra copy of chromosome 21, affects both genders. And Klinefelter syndrome leads to taller stature, reduced testosterone, and potential fertility issues. Multifactorial disorders. So these are disorders caused by a combination of genetic and environmental factors, like heart disease, influenced by genetics, diet, lifestyle. Diabetes, for example, genetic predisposition with lifestyle triggers. And we have cancer, so genetic mutations, plus environmental factors like smoking. Now let's discuss some inheritance patterns of genetic disorders. So we have autosomal dominant, where a single mutated gene from one parent is enough to cause the disorder. For example, we have Huntington's disease here. So affected individuals have a 50% chance of passing it to their children. So only one copy of the mutated gene is needed. Then we have autosomal recessive, where both copies, so 2 copies of the gene, must be mutated for the disorder to manifest, for example, in cystic fibrosis. And carriers, so 1 mutated copy, are typically unaffected but can pass the mutation to the offspring. Then we have X-linked. These are caused by mutations on the X chromosome. These disorders often affect males more severely as they have only 1 X chromosome. So, for example, hemophilia. And females may be carriers with mild or no symptoms. So, example, if one parent carries a dominant mutation, each child has a 50% chance of inheriting it, while recessive conditions require both parents to be carriers for a child to be affected. Now, how do we go about diagnosing genetic disorders? So we have different methods like genetic testing, identifying mutations like the BRCA1 gene for breast cancer. We have karyotyping, where we're visualizing chromosomes to detect abnormalities. We have newborn screening, so we test for common genetic conditions at birth. And early detection enables treatment or management. And briefly on treating these genetic disorders. So if it's symptomatic, we have symptomatic management like medications, physical therapy, or dietary adjustments. If it's gene therapy, we are fixing faulty genes or adding healthy ones. Advances are currently in CRISPR technology, so genome editing to treat genetic diseases. For example, gene therapy has been used to treat spinal muscular atrophy by replacing the faulty gene. So in summary then, mutations can be spontaneous or caused by external factors. Types of mutations include point mutations, frame shifts, and chromosomal abnormalities. Genetic disorders can be single gene, chromosomal, or multifactorial. And advances in genetic testing and therapy are transforming medicine. That's the end of Chapter 6. Continue now to Chapter 7. Thank you for listening. Welcome to another lecture by MedicoMedics, Learning Made Easy, Genetics Chapter 7, Population Genetics. So in this lecture, we are discussing population genetics, talking about genetic variation, the Hardy-Weinberg principle, factors affecting genetic variation, continuing with gene flow, natural selection, application, and end with a summary. Now, population genetics is the study of how genetic variation is distributed and changes over time within a population. The goal is to understand genetic diversity and to analyze evolutionary forces shaping populations. Why is it important? Because it helps in disease studies, in conservation, biology, and understanding human evolution. So population genetics looks at how genes are spread across groups of people, animals, or plants, and how they change over time. So what are some sources of variation? Well, it can be through mutation, where new alleles are introduced. Recombination, which shuffles genes during meiosis. And gene flow, the movement of genes between populations. Then we have the concept of genotype versus allele frequency. So genotype frequency is the proportion of individuals with a specific genetic makeup. Allele frequency means proportion of a specific allele in the population. So genetic variation comes from mutations, gene mixing, and movement of individuals, creating diversity in population. An example of this could be in a population. If 70% of people have allele A, capital A, whatever that is, and 30% have allele A, the allele frequencies are 70% and 30%. So now let's see some practical calculations. So we have the Hardy-Weinberg principle. And it is a mathematical model used in population genetics to predict that allele and genotype frequencies in a population will remain constant from generation to generation, provided that no evolutionary forces such as natural selection, mutations, migration, genetic drift, etc., act on it. So it kind of represents a genetic equilibrium. Now, for a population to be in this Hardy-Weinberg equilibrium, some conditions must be met. So we make these assumptions. First, large population size. So this minimizes genetic drift. Then we have random mating. So no preferences for specific traits. That's another assumption. Then we have no mutations. So no new alleles introduced. No gene flow, meaning no migration in or out. And no natural selection. So no allele has a reproductive advantage or a survival advantage. So these are the basic assumptions. Then we have a way to use this to identify how many percent of individuals are a certain are heterozygotes. So we have the equation here, right? P squared plus 2pq plus q squared is equal to 1. So p squared here is the frequency of homozygous dominant or homozygous dominant, so AA. 2pq is frequency of heterozygotes, AA, and q2 again, but for recessive, so AA. And here is an example of a calculation. So if p, the dominant allele, is 0.6, that squared will give us 36%. And if q, the recessive allele, is 0.4, that will give us 0.16%. So 2pq would give us 2 times 0.6 and 0.4. That will give us 0.48, meaning 48% of individuals are heterozygous. And this principle helps scientists study whether a population is evolving by comparing real-world genetic data to expected frequencies under equilibrium. Now, what are some factors affecting genetic variation? Well, we have mutation, where there is an introduction of new alleles. We mentioned before gene flow, so the movement of alleles between populations. Genetic drift, so random changes in allele frequencies, especially in small populations. And of course, natural selection, so survival and reproduction of individuals with advantage traits. So for example, if you look at genetic drift, so in a small village, a rare allele might disappear by chance if no one with it has children. So genetic drift are random changes in allele frequencies in small populations. The effect would be reducing genetic diversity and increase chances of losing rare alleles. The types are bottleneck effect, so a population is drastically reduced, leaving limited genetic diversity. Then we have founder effect, so a small group establishes a new population with limited variation. Example, bottleneck, so a natural disaster kills most of a population, leaving only a few survivors with a narrow gene pool. Gene flow is the movement of alleles between populations through migration. The effects, increasing genetic variation in a population, but reduces differences between populations. So for example, a group of individuals migrate to a new region and mixes their genes with the local population. Natural selection. The definition is differential, survival and reproduction of individuals due to advantageous traits. Types could be stabilizing selection, so favors the average trait, like human birth weight. Directional selection favors one extreme trait, like faster cheetahs. Disruptive selection favors both extremes, not the average. So for example, beak sizes in finches. So example here is antibiotic resistance in bacteria is an example of directional selection. So natural selection essentially is like nature choosing the best tools for survival. And so these tools, these traits, become more common. Some applications of population genetics. So in medicine, for example, understanding genetic predispositions to diseases. It helps identifying populations at risk for inherited disorders. In conservation biology, it helps preserving genetic diversity in endangered species. In human evolution, helps us trace migration patterns and ancestry. A medical example could be sickle cell allele persists in population where malaria is common because it provides resistance to the disease. In summary then, genetic variation is influenced by mutation, gene flow, genetic drift, and natural selection. The Hardy-Weinberg principle provides a baseline for studying populations, and applications range from medicine to conservation and understanding human evolution. So, all in all, population genetics looks at how genes change groups over time, helping us understand diseases, evolution, and even how to save endangered species. And that's the end of Chapter 7. Continue now to Chapter 8. Welcome to another lecture by MedicoMedics, Genetics Chapter 8, Cytogenetics. In this lecture, we will be discussing cytogenetics. What is it? We will talk about chromosome structure and function, karyotyping, mention some chromosomal abnormalities, some clinical examples. Continuing on, we will talk about some applications of cytogenetics and, as always, end with a summary. Now, cytogenetics is the branch of genetics that studies the structure and function of chromosomes. So focus areas are chromosomal abnormalities, techniques for analyzing chromosomes, and of course, clinical significance of genetic disorders. Now, chromosomes, structure and function. So what are they? They are structures that organize and store DNA. And humans have 20 pairs of these, 46 in total. What's its structure? Well, it has a centromere, which divides chromosomes into short and long arms. We have telomeres. This is the protective cap at the end of the chromosome. What is its function? It's to ensure proper DNA replication and segregation during cell division. Example, chromosome 1 is the largest human chromosome, carrying about 2,000 genes. So chromosomes are like bookshelves that store and organize DNA, so it's easy to find during cell division. Now, what is then karyotyping? Well, it is a visual representation of chromosomes arranged by size and shape. We use it to detect chromosomal abnormalities, for example, Down syndrome. It helps us identify gender. So is it XX or XY chromosomes? If you remember, XX is female and XY is, you guessed it, male. And Down syndrome, for example, is also known as trisomy 21, where we have an extra copy of chromosome 21. So the process of karyotyping, we collect cells, so blood and amniotic fluid. We stimulate cell division, do a stain, and photograph the chromosomes. So, as mentioned in Down syndrome, karyotyping revealed that the chromosome in the extra chromosome in trisomy 21 in Down syndrome. So a karyotype, you can think of it as a photo of your chromosomes lined up to check for missing or extra pieces. Chromosomal abnormalities, different types. So we have numerical abnormalities. I think this one is aneuploidy, and if I'm pronouncing that wrong, live with it. So this means extra. You have an extra or missing chromosomes. So for example, in trisomy 21, you have an extra chromosome. So in Down syndrome, for example, and we call it trisomy 21. If you have monosomy, you have a missing chromosome, right? And this is an example of this is Turner syndrome, where you have 45 in total. What about structural abnormalities? Well, there could be deletion. So there is a loss of some chromosome segment, like in Cri-du-Chat syndrome. You can have duplication, where there is an extra copy of a chromosome segment. You can have translocation, where segment moves to a different chromosome. You can have inversion, segment flips within the same chromosome. So having these kind of chromosomal abnormalities are like books missing pages. So having extra pages or sections swapped with some other book, it's all mixed up. That's essentially what we're talking about here. Now, what are some clinical examples of chromosomal disorders? Well, we have numerical disorders like Down syndrome, trisomy 21, and we can have the clinical features of developmental delays, characteristic facial features, etc. We can have Turner syndrome, right, where there is a missing copy. Here we have short stature or infertility in females. Klinefelter syndrome, where you have an extra copy, 47 chromosomes, and this is produces male infertility and learning disabilities. Now, Klinefelter only applies to males, and Turner syndrome only applies to females. What are some structural disorders then? We talked about numerical. What is structural? Cri-du-Chat syndrome, for example. And typically it's a high-pitched crying in infants, and you see developmental delays. Chronic myeloid leukemia caused by a translocation between chromosome 9 and 22, also called Philadelphia chromosome. So some of these disorders are caused by having too many or too few chromosomes, while others result from broken or rearranged pieces. So hopefully you have now an idea of how to differentiate between these disorders and these. It's actually on the level of the chromosomes itself. Here you have a issue with a quote-unquote broken or rearranged setup of your chromosome. Here you have chromosomes either missing or you have too many. Now, cytogenetic techniques. So we mentioned karyotyping, which helps us visualize entire chromosomes for abnormalities, like a photograph. We also have, the abbreviation is FISH, so fluorescence in situ hybridization. Here we use fluorescent probes to detect specific DNA sequences. For example, identifying chromosomal deletions in cancer. We have comparative genomic hybridization, detecting gains or losses of chromosomal regions. We have arrays, so higher resolution for detecting submicroscopic abnormalities. So, for example, fluorescence in situ, so FISH, is used to confirm the presence of the Philadelphia chromosome in leukemia. So cytogenetic techniques are advanced microscopes, essentially, that help scientists to see changes more clearly. Now, what are telomeres? So these are repeated DNA sequences at the end of chromosomes, and they help in protecting chromosomes from damage. Their significance: shortening of telomeres is associated with aging. Dysfunctional telomeres can lead to cancer. So think of telomeres essentially as a protective structure for your chromosomes. An example: cells in older individuals often have shorter telomeres, and this is linked to aging and cell death. So, yeah. Mosaicism or mosaicism, however you want to pronounce that, is the presence of two or more different genetic cell lines within an individual. So it's like having two types of cells in your body, each with a slightly different genetic makeup. Causes errors during early cell division in the embryo. Example is mosaic Down syndrome. So some cells have an extra chromosome 21 while others don't. Mosaic Turner syndrome. So some cells lack an X chromosome and others don't. So essentially it's a hybrid form of having trisomy 21 or Turner syndrome. You have both quote-unquote healthy chromosomes or cells, and you have the presence of extra copies or missing copies in other cells. What are some applications of cytogenetics? Well, in medicine, we use it to diagnose genetic disorders. For example, prenatal testing for Down syndrome. It helps us identifying cancer-related chromosomal changes. It's used in reproductive genetics, detecting chromosomal issues in embryos using IVF. In research, so understanding chromosome structure and function. For example, cytogenetics helps identify chromosomal abnormalities in embryos to improve IVF success rates. And IVF is in vitro fertilization. So cytogenetics is used in medicine to diagnose disorders, but also in fertility clinics to ensure healthy embryos, and in labs, of course, to study chromosomes. So in summary then, cytogenetics studies chromosomes to understand their role in genetics. Chromosomal abnormalities can be numerical or structural. Techniques like karyotyping and fluorescence in situ FISH are critical for diagnosing genetic disorders, and applications include medicine, reproductive genetics, and research. That's the end of Chapter 8. Continue now to Chapter 9. Welcome to another lecture by MedicoMedics, Genetics Chapter 9, Genomics. In this lecture, we will discuss what is genomics, talk about the Human Genome Project, mention some applications, some key tools, some types of genomic studies, challenges, the future, and end with a summary. Now, genomics is the study of an organism's entire genome, including all its genes and non-coding regions. Focus areas include genome sequencing, so reading the entire genetic code. Comparative genomics, comparing genomes across species. And functional genomics, understanding gene expression and interactions. What are some applications of this? Well, we use it in personalized medicine, disease research, and evolutionary studies. So genomics is like studying an entire library of DNA to understand how it works, evolves, and influences health. Now, the Human Genome Project was a global effort to sequence all 3 billion base pairs of human DNA, and it started in 1990 and completed in 2003. Key achievements: it identified approximately 20 to 25,000 human genes, and it mapped all genetic variations. The impact? Well, it laid a foundation for personalized medicine and enabled genome-wide association studies. So it was like creating a detailed map of all human genes to better understand health and disease. High-throughput sequencing, so the next generation of sequencing, is an advanced technology to sequence DNA rapidly and at a large scale. So how does it work? Well, DNA is fragmented. Fragments are amplified and then sequenced in parallel. And then we use computational tools to assemble the sequence. The advantages: it's faster, cheaper, and more accurate than earlier methods, like, for example, the Sanger sequencing. So sequencing an entire genome now takes days instead of actual years. Some applications in medicine: identifying disease-causing genes like BRCA mutations, developing targeted therapies. Agriculture: breeding disease-resistant crops, improving livestock genetics. Evolutionary biology, so tracing human ancestry and migration patterns, studying genome evolution across species. Some key tools used in genomics: we have polymerase chain reaction, PCR, where we amplify specific DNA sequences for analysis. Microarrays, where we analyze gene expression or detect genetic variants. CRISPR technology, so genome editing tool to modify genes with precision. Bioinformatics, software and algorithms to analyze large genomic data sets. CRISPR has been used to treat genetic conditions, for example, like sickle cell anemia. Some different types of genomic studies. So we have whole genome sequencing, which sequences the entire genome to identify all genetic variations. Exome sequences focuses on protein coding regions of the genome. Genome-wide association studies identifies genetic variants associated with diseases by comparing genomes of affected and unaffected individuals. Examples: genome-wide association studies studies the identified genes linked to diabetes, heart disease, and Alzheimer's. Genetic variation and its implications. So we have different types of variations. We have, for example, single nucleotide polymorphisms, where we talk about single base changes. We have copy number variations, so duplications or deletions of DNA segments. We have structural variations, so larger changes in the DNA structure. Why is this clinically relevant? Well, for example, single nucleotide polymorphisms are linked to drug responses, something called pharmacogenomics, which we will talk about in an upcoming lecture. And we have variations causing genetic disorders. An example of this is single nucleotide polymorphisms in the CYP2C19 gene affect how individuals respond to the blood thinner clopidogrel. This is directly relevant, clinically relevant. Some challenges in genomics. So data volume. So sequencing generates massive data sets requiring advanced computational tools. Ethical concerns. Privacy of genetic information. Discrimination based on genetic data. Clinical translation. So bridging the gap between research and practical application in medicine. These are some of the challenges lying ahead. Now, what does the future hold? Well, some trends are personalized medicine based on individual genomes, advances in artificial intelligence and machine learning for genomic analysis, synthetic genomics, so designing and building artificial genomes. Potentials, well, it gives us the ability to maybe eradicate genetic diseases, revolutionizing drug development, and creating bioengineered organisms for specific purposes. This might be a bit scary, but, let's see. An example, AI tools are being developed to predict cancer risk based on genomic data. So, in summary then, genomics studies the entire genome to understand its structure, function, and evolution. High-throughput sequencing technologies have revolutionized how quickly and accurately we can sequence DNA. Applications span medicine, agriculture, and evolutionary biology. Challenges include data management, ethical concerns, and translating research into practice. So genomics is about studying all our DNA, using fast and advanced tools to improve health, food, and our understanding of life itself. That's the end of Chapter 9. Continue now to Chapter 10. Thank you for listening. Welcome to another lecture by MedicoMedics, Genetics Chapter 10, Epigenetics. In this lecture, we will talk about the concept of epigenetics. What is it? Talk about mechanisms. Go into detail on DNA methylation. Talk about histone modification. And continuing to epigenetics in development and disease, in medicine, its future, and end with a summary. Now, epigenetics is the study of heritable changes in gene expression that do not involve changes in DNA sequence. Let's repeat that. Epigenetics is the study of heritable changes in gene expression that do not involve changes in DNA sequence. So the key idea here is genes can be turned on or off based on external or internal signals. Why does it matter? Because it explains how the same DNA produces different cell types. For example, skin cells versus nerve cells. And it links environment to gene activity. So epigenetics is like using a bookmark or using bookmarks to highlight certain pages in a book. And this book here is genes, right? So it's used to highlight certain pages so they're easy to read or to skip. Now let's mention some mechanisms of epigenetic regulation. So first we have DNA methylation. So methyl groups attach to DNA, typically silencing genes. Example is inactive X chromosomes in females. Another is histone modification. So chemical tags on histones loosen or tighten DNA, affecting gene expression. And histones are these proteins which DNA is wrapped. Then we have non-coding RNAs. So small RNAs like miRNAs regulate gene expression post-transcriptionally. Now, DNA methylation. So what it does is it adds methyl groups to cytosine based in DNA. Remember the pairings of A to T and C to G in DNA? We had adenine, thymine, cytosine, and guanine. So here we're talking about cytosine. So we add methyl groups here. And this silences genes by preventing transcription factors from binding. Clinical importance: abnormal methylation is, for example, linked to cancer, like tumor suppressor genes being silenced. So an example is hypermethylation of the BRCA1 gene can increase the risk of breast cancer. So the idea with DNA methylation is like putting a sticker, right, over a certain word or words in a book, and this will make it unreadable. Say you have a system reading it and then producing proteins. Following that, you hide this little part, or you use a rubber and remove it or whatever. So it becomes unreadable. Ergo, this protein that is then being the result of it will disappear. Now, the second way was to do histone modification. So here we are talking about chemical changes to histones affect how tightly DNA is wrapped. And we know that loosely wrapped DNA is accessible for transcription. So essentially to continue to produce proteins and move into translation, while tightly wrapped DNA is not. So if we can find a way to tightly wrap DNA, it will not be accessible for transcription. So examples of acetylation is it opens up chromatin activating genes. And methylation can either activate or silence genes depending on context. So histone modification is like loosening or tightening a belt. Loosening it makes it easier to use the DNA. Role of non-coding RNAs in epigenetics. These regulate gene expression without coding for proteins. For example, microRNAs, these block translation of mRNA into protein. Remember, we had transcription, DNA to RNA, and then we had from messenger RNA to amino acid chains, meaning protein production. So what this does is it blocks translation, this bit right here, from producing proteins. Now we have long non-coding RNAs as well. These interact with chromatin to regulate gene activity. Clinical example is that microRNAs dysregulation is linked to cancer progression. So non-coding RNAs are like, think of traffic signals when you're driving a car or something. They're like traffic signals that tell your genes when to stop or when to go during protein production. Now let's talk about epigenetics and development. So, as we mentioned, epigenetics refers to changes in gene expression that do not alter the DNA sequence, but affect how genes are turned on or off. Now, these modifications are crucial for cell differentiation, where different cell types like muscle, nerve, or skin cells develop from the same genetic material. This is achieved through mechanisms like we mentioned: DNA methylation, histone modification, and non-coding DNA regulation. So these can control gene activity without changing the underlying DNA sequence. And some examples could be imprinting. which is parent-specific gene expression. So some genes are expressed only from one parent's allele, while the other is silenced. For example, we have insulin-like growth factor 2, which is typically expressed only from the paternal allele, while the maternal copy remains inactive. And this parent-specific gene regulation is crucial for normal growth and development. Another one is X chromosome inactivation, where we silence one X chromosome in females for dosage compensation. For example, in female mammals, one of the two X chromosomes in each cell is randomly silenced to ensure equal gene dosage between males and females, because we both have an X chromosome. And this is occurring through the action of something called the X inactive specific transcript gene, which coats the inactive X chromosome and prevents its gene from being expressed. And this mechanism prevents an overexpression of X-linked genes in females who have two. So these epigenetic processes somehow ensures proper development, cellular function, and stability, etc. in an organism. They also play a role in disease when disrupted. Like, for example, a genetic disorder like Prader-Willi syndrome, which is caused by imprinting errors. Now then, epigenetics and disease. So, for example, in cancer, hypermethylation of tumor suppressor genes, like a tumor suppressor gene could be P53, or as mentioned several times in these lectures, is the BRCA1 gene. And hypermethylation of these tumor suppressor genes can silence their function, leading to uncontrolled growth. Hypomethylation can activate oncogenes, which promote cancer progression. Methylation-based therapies, for example, can help treat cancer by reversing abnormal DNA methylation patterns that silence these tumor suppressor genes or that activate oncogenes. So we have, for example, DNA demethylation agents like dicetabine, I think it's called, and these reactivate these tumor suppressor genes by removing excess methylation, right? Because hypermethylation of these would silence them, leading to uncontrolled growth. We have these demethylation agents that would then reactivate the tumor suppressor genes, leading to inhibiting this uncontrolled growth. So targeted epigenetic drugs can selectively modify methylation patterns to restore normal gene function. And by correcting epigenetic dysregulation, these therapies can slow tumor growth, enhance immune responses, and improve treatment outcomes. So this is offering us a very promising strategy when dealing with cancer. And we also have, for example, in neurodegenerative disorders. So in Alzheimer's disease, for example, epigenetic modifications alter memory-related genes, affecting neuron function and accelerating cognitive decline. In metabolic disorders, epigenetic changes link environmental factors like diet and obesity to diseases like diabetes. And it does this by affecting genes involved in insulin regulation and metabolism, etc. Now, some environmental factors in epigenetics. So factors that influence epigenetics include, for example, diet. So nutrients like folate or vitamin B12 support methylation. Toxins, so chemicals like bisphenol A, can alter epigenetic marks. Stress, so chronic stress, can modify gene expression. We have transgenerational epigenetics, so epigenetics changes or epigenetic changes can be passed to offspring, affecting future generations. So, for example, a high-fat diet in pregnant mice caused obesity-related epigenetic changes in their offspring. What about in medicine? So, therapeutic applications, epigenetic drugs, so DNA methylation inhibitors, like we just mentioned, and histone deacetylase inhibitors are used in cancer treatment. Personalized medicine, so tailoring treatments based on epigenetic profiles. We have these biomarkers to detect cancer and other diseases early. Like we mentioned before, aceticidine or acecitidine, however you want to go with that, is a drug that reverses DNA hypermethylation, for example, in leukemia. What does the future hold then? So we have a lot of trends in research. So we have advances in single-cell epigenomics, understanding epigenome's roles in aging, using AI for these analyses. Potentials include reversing aging-related epigenetic changes, developing gene therapies targeting epigenetic modifications. So in summary then, epigenetics regulates gene activity without changing the DNA sequence. Mechanisms include DNA methylation, histone modification, and non-coding RNAs. Environmental factors influence epigenetic marks, linking lifestyle to health. And epigenetic research is driving new therapies and advancements in personalized medicine. And that's the end of Chapter 10. Continue now to Chapter 11. Thank you for listening. Welcome to another lecture by MedicoMedics, Genetics Chapter 11, Pharmacogenetics. In this lecture, we will talk about pharmacogenetics, mention some key concepts, talk about pharmacogenetics and drug response, adverse drug reactions, continuing down to pharmacogenetics in personalized medicine, and end with a summary. Now, pharmacogenetics is the study of how an individual's genetic makeup affects their response to drugs. And the goal is to optimize drug therapy for each individual based on their genetics. Relevance: to reduce adverse drug reactions, to enhance drug efficiency, and to guide personalized medicine. So pharmacogenetics studies how your DNA affects the way medicines work in your body, like customizing a treatment plan just for you. Some key concepts are drug metabolism, so enzymes break down drugs in the body. Genetic variations can make enzymes either too fast, so ultra metabolizers, or too slow. Another concept is pharmacokinetics, so how the body absorbs, distributes, metabolizes, and excretes drugs. Another one is pharmacodynamics, so how drugs affect the body, influenced by genetic differences. So essentially, your genes affect how quickly or slowly your body processes medicines and how well they work. So some variations in how drug metabolism is affected. For example, cytochrome P450 enzymes, CyPCs. This is a family of enzymes responsible for metabolizing most drugs. Common CYP genes include, for example, CYP2D6. It affects painkillers like codeine and tramadol. Another one affecting blood thinners like clopidogrel. CYP3A4 metabolizes many drugs. What is the impact of these variants? Well, poor metabolizers increase drug toxicity. Ultra-rapid metabolizers will reduce drug efficiency. For example, codeine is converted to morphine. Codeine is converted to morphine by CYP2D6 enzyme. And poor metabolizers experience little pain relief, while ultra-metabolizers risk overdose. So some people process medicines too slowly or too quickly because of their genes. So this can make drugs either too strong or not work at all. Now let's briefly mention a little bit of how genetic testing could help us adjust medications to make them safer and more effective. For example, warfarin, which is a blood thinner. Depending on these genes, the variation will affect how the drug is metabolized and their sensitivity to it. So we can adjust doses based on their genetic setting. When we talk about antidepressants, so if you have the gene SLC6A4, which is a serotonin transporter gene, the variations of this gene will influence the response to serotonin, to selective serotonin reuptake inhibitors like fluoxetine. In cancer drugs, for example, you have the gene TPMT, and variations in this can affect tolerance to drugs like mercaptourin. Now, adverse drug reactions are unintended, harmful effects of medications that occur at normal doses used for treatment. And they range from mild side effects like nausea to severe and life-threatening reactions like organ failure or anaphylaxis, etc. So certain genetic variations affect how individuals metabolize, respond to, or tolerate drugs, leading to a higher risk of these adverse drug reactions. So, for example, the HLA-B gene, which plays a key role in our immune system recognition, some variants predict hypersensitivity reactions. For example, we have HLA-B 5701, right? And this is linked to severe allergic reactions to the drug abacavir, which is an HIV drug. And this causes hypersensitivity syndrome to this drug, which can be life-threatening. Another example is the CYP2C9 variant or enzyme. So this enzyme is a liver enzyme that metabolizes warfarin, which is a blood thinner. And the variants, different variants of this, like CYP2C92 or 93, slow metabolism, which increases bleeding risk due to excessive drug accumulation. So understanding genetics, genetic links to these adverse drug reactions allows for very personalized type of medicine, where drug choice and dosing can be tailored to an individual's genetic profile, which would, of course, improve safety and efficiency. What about pharmacogenetic testing? So what is it? It's a genetic test to identify variations that affect drug response. How it works is we collect a DNA sample from blood or saliva. We analyze genes associated with drug metabolism and response. The application, naturally, we can tailor treatments for cancer, heart disease, mental health, and more. For example, genetic testing for CYP2C19 can guide the use of clopidogrel in patients at risk for heart attacks. Now, as we've mentioned, personalized medicine is tailoring treatments to an individual's genetic profile. Some key benefits include fewer side effects, better efficiency of the drug, improved patient outcomes. So the idea is to use your specific genetic information to give you the best treatment possible, like building a custom-made suit. Some challenges in pharmacogenetics include scientific issues like limited understanding of rare genetic variants, the complexity of the gene-environment interaction, ethical concerns like privacy of genetic information or risk of genetic discrimination, and of course in clinical implementation, so the cost of genetic testing and integrating genetic data into medical practice. So it has a great potential, but challenges like cost and privacy, etc., need to be addressed. What does the future hold? Where upcoming trends include combining genetic data with AI to predict drug responses, expanding pharmacogenetics testing to more populations, developing more targeted therapies. Potentials include transforming healthcare with a safer, more effective treatment, reducing healthcare costs by preventing these adverse drug reactions. Example could be AI tools used to analyze your entire genome to recommend the best drug treatment for you. So in summary then, pharmacogenetics explores how genetic variation affect drug response. Genetic testing helps tailor medications to individuals, improving safety and effectiveness. Applications include cancer therapy, heart disease treatment, and managing mental health disorders. Challenges include ethical ones, cost issues, integrating genetic data into healthcare systems. So pharmacogenetics is about matching the right medicine to the right person using DNA, making treatments safer and more effective. That's the end of this lecture. Continue now to Chapter 12. Welcome to another lecture by MedicoMedics, Genetics Chapter 12, Cancer Genetics. In this lecture, we will talk about cancer genetics. What is it? Talk about different types, key genes involved, differentiate between hereditary and sporadic cancer, mention some examples, talk about some risk factors, talk about the future of cancer genetics, and end with a summary. Now, cancer genetics is the study of genetic mutations and alterations that contribute to cancer. We know that cancer arises from uncontrolled cell growth due to genetic changes, and both inherited and acquired genetic mutations can contribute to this. Its importance, therefore, is to help identify risk factors and guide the development of targeted therapies. So in cancer genetics, we are studying how changes in our DNA can cause cells to grow uncontrollably, leading to tumors. Some types of genetic changes in cancers. Well, first, mutations. These are changes in the DNA sequence, and they could be point mutations, insertions, or deletions. If you look at previous lectures, we've mentioned these in detail. Another thing could be chromosomal alterations, so deletions, duplications, translocations, like Philadelphia chromosome in leukemia, also talked about in previous lectures. And of course, epigenetic changes, so altered gene expression without changes in DNA sequence, for example, DNA methylation. Some key genes involved in cancer. So we have something called oncogenes. These genes are called oncogenes because they promote cell growth and division. Mutations in these cause them to be overactive. For example, HER2 in breast cancer. We have tumor suppressor genes. These are genes that prevent uncontrolled growth. And mutations can deactivate these tumor suppressor genes. Examples of these suppressor genes are TP53, BRCA1, BRCA2, etc. DNA repair genes. These help in repairing and fixing damaged DNA. And mutations here increase genetic instability. Examples are MLH1 and MSH2 in Lynch syndrome. So all in all, some genes act like gas pedals in a car, like oncogenes. You have tumor suppressor genes that act like car brakes, and you have repair tools. So cancer typically happens when these different systems fail. Either one fails here and this one becomes overactive, or this one doesn't work and the reparation doesn't work, etc., etc. So it's a combination of these key factors. Now, hereditary cancer is caused by inherited genetic mutations. It accounts for approximately 5 to 10% of all cancers. Example, BRCA1 and 2 mutations in breast and ovarian cancer. Then we have sporadic cancer caused by acquired mutations during life. This accounts for approximately 90 to 95% of all cancers. And the factor here are environmental exposures like smoking and UV radiation, etc. So hereditary cancers are passed down in families, while sporadic happen due to life choices or exposures, etc., etc. Now then, let's mention some examples of hereditary cancer syndromes. So we have BRCA-related breast and ovarian cancer, where we have mutations in the BRCA1 and 2 gene, increasing the risk. This follows a autosomal dominant inheritance pattern. Then we have Lynch syndrome, mutations in the DNA repair genes, like MLH1 and MSH2. And this increases risk of colorectal and endometrial cancer. We have familial adenomatous polypses. This is a mutation has occurred in the APC gene and leads to numerous colon polyps and colorectal cancer. Now let's briefly mention the hallmarks of cancer. We have a course dedicated to oncology that you can check out, where we go into all of these things in detail. But here we can just mention the key points. So we have sustained cell growth. We have another one in the hallmark of cancers. We have evading growth suppressor genes. We have resisting cell death, inducing blood vessel formation, enabling immortality, invasion, and metastasis. So cancer cells are like rule breakers. They grow uncontrollably, ignoring, for example, these stop signals and spread to other parts of the body. Some cancer risk factors could be genetic, so inherited mutations like the BRCA1 or 2 gene. Indeed, environmental, like UV radiation, tobacco, carcinogens in general. Lifestyle, diet, physical activity, alcohol consumption. They can be viral, so like HPV is linked to cervical cancer. Hepatitis B and C is linked to liver cancer. We have targeted cancer therapies. Now, what are these? These are drugs that are specifically targeting genetic mutations in cancer cells. For example, HER2 inhibitors. We have EGFR inhibitors. For lung cancer, we have PARP inhibitors for BRCA mutated cancers. The advantage here is that it's fewer side effects compared to traditional chemotherapy. So targeted therapies are like very precise tools. They are precision tools that attack cancer cells without harming healthy ones as much. So the purpose for genetic testing for cancer could be, for example, to identify individuals at high risk for hereditary cancers. It can help guide personalized treatments. Tests, for example, for BRCA1 and 2 could give us information for breast and ovarian cancer, Lynch syndrome panel for colorectal cancer. And the implication of this is early screening and prevention strategies. And of course, family counseling for at-risk relatives. Now, cancer prevention and early detection. So prevention, you can do lifestyle changes like quitting smoking, healthy diet, exercising, vaccinations like HPV vaccine for cervical cancer prevention, avoiding environmental carcinogens. Another one is early detection, so regular screenings like having your mammograms done, colonoscopies, genetic testing for high-risk individuals. Challenges in cancer genetics. So it's quite complex because cancer is often caused by multiple mutations. Genetic testing and targeted therapies are actually quite costly and not widely available. And we always have to mention ethical issues like privacy of genetic data or discrimination risks. So the future of cancer genetics. Advances in genomics. So whole genome sequencing helps us identify rare mutations. Liquid biopsies detecting cancer through blood tests. Immunotherapy, so using the immune system to fight cancer. Artificial intelligence in cancer research, so predicting cancer risk and designing personalized treatments. Example, liquid biopsies are being developed right now to detect cancer earlier and less invasively. So in summary then, cancer results from genetic and epigenetic changes that disrupt normal cell growth. Key genes include oncogenes, tumor suppressor genes, and DNA repair genes. Genetic testing and targeted therapies improve diagnosis, prevention, and treatment. Future technologies like genomics and immunotherapy promise breakthroughs in cancer care. For in-depth detail into the world of cancer, please check our course on oncology. That's the end of Chapter 12. Thank you for listening. Continue now to Chapter 13. Welcome to another lecture by MedicoMedics. So Genetics Chapter 13, Genetic Counseling and Ethical Issues. So in this lecture, we will have a brief overview over genetic counseling. When is it recommended? What are the steps involved? Some types of genetic testing, benefits of genetic counseling, ethical issues, and end with a summary. Now, genetic counseling is a communication process to help individuals and families understand and adapt to the medical, psychological, and familial implications of genetic contributions to disease. So key roles here include assessing genetic risks, explaining genetic testing options, and support decision-making. Who provides this? Typically genetic counselors, physicians, and trained healthcare professionals. So when is it recommended? Well, situations appropriate for genetic counseling include a family history of genetic disorders, doing a prenatal screening showing abnormal results, known carriers of genetic conditions, couples planning a family with concerns about inherited conditions, and diagnoses of a genetic condition in the family. For example, a woman with a family history of breast cancer may seek counseling to discuss genetic testing for BRCA mutations. What are the steps in the genetic counseling process? We start with an initial assessment, so collecting family and medical history, assessing risks for genetic conditions. We then move on to genetic testing, discussing options and implications. Conduct tests if necessary, like blood or saliva sample. Result interpretation, so we explain what the results mean for health and family planning. Support and decision making, so providing emotional support and helping with decision about prevention or treatment. So we have several different types of genetic testing. We have diagnostic testing, so identifying genetic conditions causing symptoms like Huntington's disease. We do predictive testing, so assessing the risk of developing a condition like BRCA testing for breast cancer. We do carrier testing, identifying if someone carries a gene for a recessive condition like cystic fibrosis. Prenatal testing, detecting genetic conditions in a fetus like looking for Down syndrome. Newborn screening, identifying genetic conditions at birth. Now, what are some benefits of genetic counseling? Medical benefits include early detection and prevention of genetic disorders, informed decision-making about treatments or surgeries, emotional benefits, so reduced anxiety through understanding the risks, support in coping with genetic conditions, family planning, so it helps couples make informed choices about having children, So, for example, a family with a history of sickle cell anemia can plan for a healthy pregnancy using genetic counseling. Now, ethical issues in genetic counseling. So, privacy and confidentiality, protecting genetic information from unauthorized access. Who has the right to know family genetic risks? Informed consent, so ensuring individuals understand the risks, benefits, and limitations of testing. Genetic discrimination, preventing misuse of genetic information by employers or insurers, etc. Autonomy and decision-making, so respecting individual choices even when they conflict with medical advice. Now, genetic counseling and the future. Up-and-coming trends, so integration of genetic data into electronic health records. Advances in whole genome sequencing. Personalized medicine based on genetic risk profiles. What are some potential positive outcomes here? Well, it could prevent more genetic diseases and help people live healthier through tailored treatments. For example, in the future, genetic tests could predict and prevent diseases like Alzheimer's decades before symptoms start. So, in summary then, genetic counseling helps individuals and families understand genetic risks and make informed decisions. Ethical concerns include privacy, informed consent, and preventing genetic discrimination. Advances in technology and personalized medicine are shaping the future of genetic counseling. Thanks for watching this lecture from MedicoMedics. If you found it helpful, don't forget to like, subscribe, and check out our other full series on microbiology, infectious diseases, and more, all right here on the channel.